So, hey everyone, welcome to the Artificial Discussion. This is our monthly podcast or webcast, about 30 minutes long, where we hope to help you demystify the digital lab or whatever a digital lab means to you or our guests. So today I'm joined by Eli Fine, who is the Director of Automation and Process Engineering at Resilience, and we'll be talking about enhancing the lab infrastructure with digital lab platforms. And so to get us started, I'm going to just turn it over to Eli to give a brief introduction, and then we will dive in. Eli?
Thanks, Ginger. I've been working in the lab automation field for a while, largely in the startup space. I've been involved in since it's been the startups, I've often been wearing multiple hats, doing some lab automation, leading some teams, doing some data science, and also some software engineering. So I've had a lot of chance to work with different aspects of and skill sets of that are involved in making things digital inside the laboratory space. So done some things with iPSC, human iPSC cell culture that involved a lot of custom scheduling. Did some work at another company with creating 3D tissues, so automating casting those tissues, and also leading the team to create some software for a new instrument we were developing to be able to read that out. And at the moment, I'm at Resilience, where we are trying to revolutionize the way that complex medicines are manufactured. And my team's role in that is helping to automate the activities that are going on in the laboratories to enable scientists to answer more questions faster with higher quality results.
Awesome. Thanks for being my guest today on the artificial discussion, and being willing to chat about this. Before we get started, since everyone has a different definition of a digital lab and what that actually means, just to get us all grounded in the discussion, what is your definition of a digital lab?
Sure. So when I think about it, I try and frame any problem on focusing on what are the specific benefits trying to be delivered, and try whenever I can to avoid just using buzzwords, although sometimes that does get you needed attention. So a couple of the things that I think are big benefits I try and focus on when we're building tools for the laboratory is trying to make sure there's observability of what's going on, not having log files that just live on an instrument and are hard to get to, but making sure that that's easily accessible in dashboards, automatically going into the cloud, things like that. So I mean, this can help us troubleshoot problems, and it also helps ensure more data-driven decisions around CapEx and helping ensure we've got optimal equipment utilization. Another big thing I like to focus on is trying to create single sources of truth. So like far too often people are when they first start out, they're copy pasting files and data all over the place, or just trying to walk up into the lab and tell a machine to run based off of their recollection of what's supposed to happen in the experiment. So trying to set up a paradigm where instruments, whether it's an analytical instrument or a piece of robotic liquid handler, are reading their instructions directly from the electronic lab notebook, and then we've got in place automated data pipelines to post the results back there, so we can set up that ELN as kind of a single source of truth. And the other key area I like to focus on when trying to digitize things is to reduce hotspots for human error. So I mean, having a single source of truth is a key contributor to reducing chances for human error, but lots of other things, taking the wrong plate out of the incubator at the wrong time, key pipetting steps that are better handled by a liquid handler, all those sorts of things, but focusing in on those hotspots and trying to knock them out in priority order.
It was funny. I was reading the other day about how many errors humans actually make in an hour, because I wanted to figure out statistics, or like when you get into numbers, right, and kind of geek out on data a little bit. Humans make a lot of errors, like for all the thought that we think that we are perfect and infallible, right? Like, so when you look at reducing hotspots for human error, like how often do you actually see errors occurring, like in the lab, if you hadn't put this infrastructure in place?
That's one of the things that we have a challenge quantifying, because if there's no system capturing the problem, then it is hard, at least I've found, to get real concrete data around those errors. I mean, you can, if there's some really weird aberrant result, people will kind of trace that back and potentially, test their pipettes and realize that they were out of calibration, or that somebody copy pasted the wrong column when they were doing the data analysis. But what I've seen happen most often is just like, it's not worth finding out why this result is so weird. We're just going to rerun the experiment. Uh, so which things were attributable to human error versus, a faulty reagent lot are somewhat more challenging. Usually, at the moment at least, we're relying largely on anecdotal information from scientists that like, hey, this is an area where there's lots of problems. Or when my team looks and analyzes a workflow that's already been identified as a good candidate to try and introduce some level of automation to, we shadow them and look for, common potential problems, small volume pipetting, lots of plates and making sure to pick out the right one. Those types of things are one area we focus on.
Okay. So I guess like kind of leveraging off of that, like how do you approach the lab infrastructure and like what best practices do you bring in for automation? Like different ways of tackling the same problems.
Sure. So one thing that I gained from my experience in these smaller companies doing some software engineering work, leading teams with some software developers on them, is I've realized that what we're doing as automation engineers in many ways is software development. Sure, we have to pick out the right instrument for the job. There's certainly like physically teaching a piece of labware on the deck has no real analog in software development, but we are installing applications on computers. We're making sure that those are functioning correctly. We are programming pieces of code that are interacting with limbs, giving instructions to robots, interfacing with schedulers. And that is very similar to what software engineers do. And there's a whole host of best practices that the software engineering community has developed that that aren't just vanity metrics. So these are things that help them, produce their code faster, help ensure that there are fewer bugs and problems that actually happen with their applications, and also make sure that, there's sufficient logging and the right metrics being collected so that if there is a problem, it's much easier for them to find out what the problem is and be able to solve it. So that at a high level is what I've been trying to do, is bring some of the software engineering best practices into the realm of R&D laboratory automation.
Does that then go and touch back in on like what you talked about from a digital lab, or when you look at that in terms of observability, that single source of truth, right? And the data-driven decisions? Does like these practices or processes that you've brought in from the software world help with that?
Sure. So yeah, I mean, we're using Git version control to make sure we've got single sources of truth. We have automated checks to ensure that the right branch from Git, essentially the exact right versions of the files, are the ones that are running when the scientists are using the instrument. We've got deterministic automated processes to go around and install the necessary applications to control the liquid handlers interface with AWS, Benchling, et cetera, on the computers, so that this is not something that we have to worry is something that might be different from computer to computer. We know that they were all installed with this exact defined procedure. So single source of truth in that sense.
Very cool. And so it's for me, I mean, I've been in the lab automation industry for a long time, right? But always on the vendor side, and worked with true or traditional lab automation engineers. It sounds like what you're employing here with like the software, engineering practices are kind of new, right? In the lab automation space, or new-ish. And so I'm wondering about like the mindset shift, right? Or was it challenging to shift the mindset of like automation engineers into this like software process, perspective and way of doing things?
Sure. Yeah. So there are automation engineers on my team who came into this with widely different levels of experience with these types of practices. Fortunately, I also have we've called informatics engineers on my team, so they've helped build some of this necessary scaffolding. One thing we encountered early on was that some of the automation engineers kind of knew how to use Git version control, but when they were typing in the commands for it, they were making some mistakes. And we had kind of an aha moment where we're thinking to ourselves, wait a minute, we're automation engineers, we're automating things for the scientists so they don't have as many manual steps to make mistakes. Why are we letting ourselves make mistakes by manually typing in all these commands? So one of the informatics engineers on my team created a little wrapper around Git that essentially just gives people, the automation engineers, three choices to walk through, so that there's no chance for them, mistyping the command or doing the wrong workflow. So that really helped make sure that the automation team was able to use Git much more easily. Let's see the other part of your question. Yeah, in terms of getting used to this, yeah, part of it was painting. I mean, we're at a startup resilience as well, so we are building things from scratch. So there was also a little bit of question early on, like when are we going to see the payoff from adding all of these checks and balances to our process? But I think one time, or there's a couple instances where it really started to click and got people on board. One time, something was about to, a file change was about to be approved to go into the main code base, but the way Git allows you to see that is it lets you see the changes to each individual file. And what someone was able to catch was that one of the engineers had accidentally changed a labware definition. And well, I mean, not accidentally changed it, but it changed it to test something and had forgotten to change it back. And so we were able to see that because we were shown very clearly the exact changes that were about to be made on a line by line basis. And that really made this like stand out. And so someone was reviewing this and caught it before it went into the main code. And I can only imagine how many times many people on this call have experienced, someone making a change to a file, things start breaking, and you realize that when you finally figure out what happened, that someone accidentally changed the labware definition. So that was an early moment that was really very exciting, seeing the payoff of using some of this. Is that saved us, untold hours of troubleshooting and also, failed experiments going on the robotics in the lab. And another tool that we created. So there's this concept called code linting in software engineering, where it's this automated checker that looks through all of the code you're creating for common mistakes that aren't necessarily a syntax error, like the code would compile and execute, but it's something that looks unusual and probably you didn't mean to do it. You might have, but probably you didn't mean to do it. And so what we did was we created a tool, again with the informatics engineer, a tool like that to be able to analyze some of the code that's controlling the robots. So to check for things like, you picked up 8 tips, but you're only actually aspirating with 7 of them. Is that really what you meant to do? Maybe, but it'll throw up a warning, and then you can add a little note in the code saying, essentially telling the linter, yes, I did truly mean to do this, and this is why. I know this is unusual, and that also helps the person reviewing the code understand your thoughts. So this was something that we presented at SLAS earlier this year, and we are just in the process of dotting all the I's and crossing the T's with legal to be able to open source this, because we think it's something that will benefit the community and hopefully other people will help contribute to, because I'm sure there are many other common liquid handler programming mistakes that we haven't yet thought of that people have ideas for to create rules that catch these types of things. Even things like one liquid handler we use, you need to manually set the tip eject height. And if you're ejecting into waste versus back into a tip box, you need to have it be a different height. And we were encountering lots of accidents where we would catch them when we were first doing, movement tests in the lab. But now this just gets caught right in the initial programming phase before you can actually commit it into our Git version control. It'll pop up a warning saying, hey, you're ejecting into waste, but you're using this tip eject height, which is different than our standard practice. And this is something we also made configurable because every team may be slightly different. Things like how you want to name your variables, how you want to name your labware, but helping to also ensure consistency across the different programmers on the team. That's another thing we've brought from software engineering is we're trying to not be in the paradigm where you assign one automation engineer to one project and just keep it kind of somewhat siloed there, but really make sure that we can have, we're all together as a team focusing on the same project at the same time to try and get it over the finish line as quick as possible. So trying to take some of that agile product development mindset that came from software engineering and apply that to the the work we're doing in the lab.
That's really cool. A question about like the code linting and in the way that it, helps check your processor in the code. Do you have to have written the liquid handling protocols in code, or is this using the liquid handling software, like the native software?
It's using the native software. So it checks the code from the native software. So some liquid handler manufacturers have made a decision that their code will be in a proprietary unreadable format by anything other than their software. And so we don't have a way in mind to make it compatible with some of those manufacturers that I won't name right now. After we open source it, if other people in the community have ideas to be able to somehow crack open those files so they can be analyzed, that would be great. But at the moment, we're using it for liquid handler families that have their programs saved in a text readable format like XML, for example.
Okay. So like I could use X liquid handler like software, write my protocol, and then run this code linting program and then it will check to make sure that what I'm doing makes sense and in a relative fashion. That's really cool.
Yeah.
So I guess it kind of touches on then, because it sounds like what you're doing is novel, at least in your approach, right? Like, and so I'm curious, like, especially for the folk on the call who are, maybe listening to being like, well, maybe like they must be doing some crazy things, right? In the lab. So I'm curious, like, what type of processes, because you mentioned liquid handling and liquid, everybody I think on this call would probably has a liquid handler experience with liquid handling at minimum, right? But what type of processes, you don't have to get into the science, but like what type of processes are you and your team automating and employing these processes and practices onto?
Yeah, so the types of top priority requests coming from the resilient scientists are probably not that much different than what you'd see at any biotech place. I mean, we have some groups where we're automating some E. coli culture colony picking. We're also automating some qPCR, digital PCR assays. Some mammalian cell culture. Some flow cytometry staining. These are what my team is doing right now from a automation perspective. The types of assays we're automating are fairly standard, I think, across the community. So I think this can be applied anywhere. One thing is resilience does have an approach of trying to make sure that we're creating biomanufacturing in a very digitally secure way because the U.S. government considers this a potential target for threat actors to try and disable the U.S.'s ability to manufacture the medicines that its citizens need. And so one thing that also a little bit kind of forced us into some more software engineering practices is this security posture that resilience has is much stricter than anyone on my team has come from before. So making sure that we're using infrastructure as code to create any resources we have in AWS. Least privilege policy. We're making sure there's no hard coded API keys or passwords saved to anywhere on the local computers. Things of that nature. That kind of forced us to go down this path a little earlier than we might have chosen at a startup where we're trying to get things off the ground. But I also don't think that you necessarily have to be operating with that strict of a security posture to benefit from these general principles. You can still do things like use version control while still hard coding your Git credentials. I wouldn't personally recommend it, but that's just a security question. That's not a question of how this can impact your ability as to make sure that the lab's operating more efficiently.
That's helpful, because one of the questions I was going to ask was like, given everything that you've implemented and you and your team have implemented at Resilience, in terms of like these software processes, software development process or software engineering processes and practices within the lab automation space, was like, is there a too soon, right? Or deployment of some of this infrastructure to benefit the lab? I can imagine if you're early days, it doesn't make sense to do it.
I mean, I would say when you're one person, it might be too soon. But as soon as you start having multiple people on a team, and you're trying to collaborate together, then having things like version control really becomes critical. And when you're one person, certainly you can benefit from things like, it automatically checking your code, but that's a lot to stand up before you've actually deployed your first method to the scientists and, have your robot doing something productive. But I would say that a lot of this really, it's certainly beneficial even for a single automation engineer. I know there's lots of companies where the automation team is just one automation engineer. It can benefit that for sure, but that's a much more nuanced consideration of your ability to execute right now versus building for a more robust future. But I would say as soon as you have a team with two people, then this is really a game changer in your ability to catch mistakes, work together effectively as a team. Another thing we've brought from software engineering is creating design documents that also live in our version control. So they kind of outline at a medium level. It's more than just process the plate, but it's not like loop 4 times and use these variables. So it can kind of fit on not a single page, but it's a single wide, and then you scroll a little bit to describe our processes. So that's really helped make sure that the team we can do these handoffs of say, okay, you're going to work this week on like this part of the process, this other person's going to work here. It also helps us show earlier to the scientists, like, this is based off of what you described to us. This is the design we are thinking of for putting this onto automation. Do you see potential problems here? And it's also been really useful for us when we're trying to troubleshoot something as we're shaking down a process. Someone says, hey, when we put these samples on, the mixing didn't look quite right. And we look at that step in the it also immediately helps orient the whole team to exactly what we're talking about without any ambiguity, because it's not like, hey, this mixing step with such and such material. It's like, no, everyone, we're screen sharing. It's this part in our procedure right here, and we can see the little notes that we've written there about the key things we did in that step. And someone pointed out, oh, we're using a slightly different tip loading procedure than we normally do for that step. That's probably why we encountered an issue with the height being different. And then sure enough, that was the problem. So it's really made troubleshooting problems different, or much easier, as well as getting the whole team quickly focused on being able to work together and trying to apply multiple people to the same problem to get things over the goal line faster, rather than having kind of slow progress independently.
Very cool. No, I really appreciate that. And I know we have a few minutes left. It wouldn't be an artificial podcast if I didn't shamelessly plug artificial along the way. So if you're not like Eli and you don't already have a lot of this great infrastructure in place, which I still think he could benefit from artificial, we do have a digital lab software platform that does some of what Eli talked about today in terms of just even providing a single user interface, on the front end of all processes to really be the glue that holds everything together, as well as digitize SOPs in really an end-to-end workflow orchestrator for the lab. That's my quick shameless plug. I'll turn it back to Eli if you have any like last minute kind of words of wisdoms around enhancing just a lab infrastructure. It sounds like, you did touch on like observability, data-driven decisions, right? So having single sources of truth and reducing like hotspots for human error. But is there anything else you want to kind of leave with folk for like any key wisdom before we close out?
Um, just one thing I first just wanted to say is I don't really see that there's overlap. I feel like tackles an entirely separate set of problems. Artificial doesn't involve the people actually creating the lines of code for the liquid handler. It doesn't go around installing the software you need on the machines for you. It does help address a lot of the things like reducing hotspots for human error, observability, single sources of truth. So I see those as the two complementary approaches. I don't think that they're really even much overlap. And both of those support those kind of three goals that I think of when trying to create a digital lab, like what is going to be the impact to the lab and to the scientists of digitizing it.
Awesome. Folks, we do have like a couple minutes for questions. If anybody has a question, feel free to pop it into chat. And if we don't get to the questions, then we would go ahead and just have Eli write them up. And when we publish this out, we would go ahead and just have him answer it then. But otherwise, Eli, I really appreciate your time and for being a guest on the artificial discussion today. I think this was a really great discussion. I learned a lot just even about software processes and how you are overlaying those into the lab automation space. So I definitely appreciate your time, and thanks everybody for joining today. And we'll see you next time.
So, all right. Thanks, Ginger.
Thanks. Have a great day. Bye, everyone.
You too.